Please make sure you sign up on the Center for EvMed website to get our weekly updates and also to RSVP for lunches. Thank you.
Okay, thank you so much. So my name is Heini. I joined SEM as a postdoc a little bit over a year ago, and I'm involved with a few different cancer-related projects. Can you hear me okay, by the way? Perfect. Today, I want to talk a little bit about a project that we're working on in collaboration with Nick Panovich from TGen. So the question that I'm ultimately interested in answering is, why do some people get cancer and others don't? And why do some cancer patients' disease keep regressing despite treatment and others respond to treatment very well? So it has already been established that some of these inherited germline genetic variants affect your risk of getting cancer during your lifetime. So you may have heard of some of these rare high penetrance risk alleles like variants in the BRCA genes that affect your risk of developing breast cancer. Or as an even more extreme example, the Li-Fraumeni syndrome, which is an hereditary cancer syndrome. So these patients have particular inherited germline variants in their TP53 tumor suppressor gene, and they're in approximately 80% risk of developing cancer during their lifetime. So these are very high penetrance, rare risk alleles. They exist in the population in a very low frequency, and a large fraction of the individuals with these variants develop cancer or show the phenotype. On the other hand, these GWAS or genome-wide association studies have identified hundreds of moderate and low penetrance risk alleles that exist in the population with a higher frequency, and only a small fraction of individuals with these variants show the cancer phenotype. On the other hand, in the recent years, several studies characterizing different somatic alterations in tumors have found these somatic mutations and copy number alterations that are associated with different cancer etiologies and disease progression and outcome. All these somatic alterations happen or occur on the background of that existing inherited germline variation, but in most cases, these processes are studied kind of independently from each other. And it's unclear how these risk variants actually affect your risk of developing cancer or the disease progression. So the question one may ask is, what is the mechanism of these cancer risk alleles, and how can we infer those mechanisms? If you look at the results from GWAS studies, many of the risk alleles are located on the non-coding parts of the human genome. Most of the mammalian genome is non-coding, and the expression of these protein-coding genes is controlled by regulatory regions that are outside those protein-coding regions. Now, if there's if there's a genetic variant or a somatic mutation in this regulatory region, it might affect the binding of these activator proteins on that regulatory region, and that can affect the expression level of the target gene. So instead of changing the actual protein product of this protein-coding gene, these variants contribute to the phenotype by changing the expression level of or how much of that protein product is being produced. So this is obviously a very simplified illustration of how transcriptional control works. And the human genome has been annotated for these putative regulatory regions, mostly based on like patterns of chromatin structure in different cell lines. But it's still very much unclear which regulatory regions are associated with which target genes. Also, these interactions can be very different in different tissues and under different conditions. And it's not very well understood how these regulatory regions or regulatory variants might contribute to cancer. My hypothesis is that the cancer phenotype is a result of inherited germline variants, acquired somatic alterations that together can affect gene expression and contribute to this cancer phenotype. And we've been exploring these topics by analyzing data from patients with multiple myeloma. If we can better understand these interactions, we can, first of all, we can better predict who will get cancer. We can predict how they will respond to different types of treatment, and we can find this transcriptional mechanism explaining or connecting the risk variants to the cancer phenotype. Multiple myeloma is a type of blood cancer that affects the plasma cells in the bone marrow. In a healthy individual, when a B cell detects an infection in the body, it matures into a plasma cell, and this plasma cell starts to produce antigens or immunoglobulins that help the body to attack that infection. But in myeloma, these plasma cells turn cancerous and they start to grow out of control and replace the cell population in the bone marrow. And these myeloma cells produce an ineffective abnormal antibody or M protein. The myeloma occurrence and mortality varies between groups with different germline genetic backgrounds. So men, and especially African American men or men with an African ethnic background, are in a higher risk of developing myeloma, and their mortality is also higher. So some of these differences between the groups could be because of environmental effects or lifestyle effects or things like differential access to healthcare. But there's also some evidence that there is a germline inherited factor affecting the myeloma risk. There is a few GWAS studies that have found a handful of myeloma risk alleles, and if your family member has myeloma, you're also in a slightly increased risk of developing myeloma. So we believe that at least some of these differences are because of these different germline effects between the groups. Data we analyzed was collected as part of the Multiple Myeloma Research Foundation's COMPASS study. So this is a still ongoing longitudinal study where the ultimate aim is to understand how the patient's genetic makeup affects the disease progression and the patient's response to treatment. This is actually the largest cancer genomics project of any single cancer in the world. And already more than 1,000 patients have been enrolled in this study. These patients are tracked for a minimum of 5 years, and new tissue samples are collected at the beginning of the study before the patient receives any treatment, and during the study, and at relapse. So currently, we're focusing on analyzing data from the baseline, so before the patient has received any type of treatment. A bunch of work has already been done on this COMPASS data, and it has led to this characterization of different myeloma subtypes based on the somatic alterations. And also, the somatic mutation burden is associated with poorer overall survival in this COMPASS cohort. I'm mainly interested in how these inherited germline variants might contribute to gene expression and through this intermediate phenotype contribute to the myeloma phenotype. So this is really all about genotype-phenotype correlations. I've been using an integrated analysis of whole genome and whole transcriptome data to look at these associations. So we have we called germline and somatic variants from whole genome sequence data, and we have gene expression levels derived from whole transcriptome sequencing. And I use expression quantitative trait loci mapping, or EQTL mapping, to find how these germline variants affect gene expression levels. So these methods are based on linear regression. So basically, we're testing for association between a variant genotype and gene expression level. And for now, we're focusing on proximal or cis effects. So we're limiting the variants that we're testing for to be within 1 megabase of the gene. Also, we're incorporating some clinical and survival data so we can test if these regulatory variants or these target gene expression levels affect survival in the COMPASS cohort. We stratified these analyses based on sex and ethnicity so that we can detect possible differential effects between these groups. Now, this figure shows the overlap of genes that were found to be under germline regulatory control in the different groups. So basically, the information contained here is the same that would be in a Venn diagram with 5 different groups, but in a slightly less anxiety-inducing format. So these are the numbers of genes that were found in each group. In the dot matrix are all the different intersects, and these are the numbers of genes in each intersect. So for instance, we found 44 genes that are under germline regulatory control only in males. Now, some of these sex or ethnicity-specific regulatory effects could be a product of just differential power to detect associations when the numbers of samples or variant allele frequencies are very different in different groups. But we did also find instances where the variant had very similar allele frequencies but different effects between the groups. So this is an example of a sex-specific EQTL. So the variant, which is located in the X chromosome, significantly affects expression of the target gene in male, but not in female or in the combined analysis of both sexes. This is because this is located in the X chromosome, the male only has like 2 possible genotypes here. So this kind of group-specific EQTLs or differences in the regulatory effects between groups could help explain these biases that we see in cancer occurrence and in mortality between the sexes and between ethnicities. In these initial analyses, we also found a handful of variants that alter gene expression and are associated with survival in this cohort. So this is one example of those. The alternative allele in this locus is associated with higher expression level of the target gene, and individuals with a genotype that has 2 alternative alleles have significantly lower survival in this cohort. So this Kaplan-Meier plots shows time on the x-axis and survival probability on the y-axis, and it basically shows the proportion of individuals in these different groups that are surviving these many days since baseline. So genes and variants like this are very interesting when we're thinking about targeted therapies and personalized medicine. So perhaps individuals that have this particular genotype would benefit on a treatment regime that targets a network of genes that this target gene is involved with. So just to summarize, we found these germline genetic variants that modulate gene expression in multiple myeloma and also are associated with survival in this cohort. Now I'm interested in analyzing this data to find how the germline genetic background might influence the architecture or landscape of somatic alterations. So how does your genetic background affect where and what type of somatic mutations or alterations happen? We're also doing somatic EQTL analysis so we can detect how these somatic alterations affect expression levels. And we're doing co-localization analysis to connect our EQTL hits to these previously published GWAS myeloma risk loci. So by doing this, we can find this intermediate phenotype or gene expression level as an explaining mechanism explaining how these risk variants lead to myeloma or how the risk variants affect the disease progression or outcome. So if we can better understand these interactions, we can better predict who will get cancer, and we can design better treatments for these people. I want to thank all my advisors, and I want to give a shout out to Austin and Bianca, who are also working on these computational analyses. And of course, I want to thank Center for Evolutionary Medicine for supporting my research here. And thank you all for listening.
Thank you very much. We have time for some questions.
For the target gene that you found, is the function of the protein of that target gene known, and does it make any sense in terms of cancer progression?
So are you?
The one on chromosome 5. Yeah, this one. So
yeah.
the ZNF.
So, yeah, this gene is actually a transcription factor. So the expression of this gene probably affects the expression of a bunch of other genes. And that's actually very interesting because so these regulatory variants can have very wide effects on top of regulating this one gene. If it acts as a promoter for another gene, for instance, it can regulate a whole network of other genes.
Yeah, good, thank you.
So what's the average age of onset for multiple myeloma?
I'm not sure actually, but it's more common in like older individuals. This is
So well into like selection.
yeah,
pseudo.
yeah, yes.
Interesting. Other questions?
I just wondered if what's known, if anything, about why African individuals have higher susceptibility.
I think at least partly it's because of the differential access to healthcare, and it's actually African American. So in this data set, there's no like African individuals. Everybody or all of those individuals were African American. But I believe there also might be these differential regulatory effects that can explain at least some of that bias.
Alrighty. Well, thank you very much, Dr. Natri. And let's... And please join us next week. We're going to be having a lecture from Mayo Clinic physician and researcher Matish Barad, who's going to be talking about hepatocellular cancers.